Hello and welcome back to our module on skeletal muscle. Today we're going to be exploring the skeletal muscle physiology. That is precisely how the skeletal muscle contracts. You'll recall previously that we looked at the arrangement of muscles themselves and how a muscle cell is made up. So let's take a quick review so that we understand exactly where we are. First of all, for example, the pectoralis muscle. The pectoralis muscle is composed of many different fascicles, and each fascicle, again, is a bundle of muscle fibers. The muscle fibers themselves are muscle cells. Once we look inside the cell, we'll see an array of proteins that are arranged themselves in myofibrils. Those myofibrils are then even more highly arranged in sarcomeres, and you'll recall that the sarcomeres are composed of actin and myosin filaments. So today, we're going to jump right into the very nitty-gritty of the actin and myosin filaments and how they interact with each other in order to effect shortening of the muscle cell itself. So first of all, let's take a quick overview of the three stages that we'll consider in muscle contraction. First of all, we'll look at the neuromuscular junction. In the neuromuscular junction, this is where we're having the signal that's come from the brain in a motor neuron to the muscle cell itself. Remember, each muscle cell has one neuron innervating it. This neuron will deliver a signal, and the signal will move along the muscle cell membrane, or the sarcolemma. And then it will be propagated throughout the cell using the system of T-tubules that we took a look at in our previous lecture. The T-tubules are tightly associated in a triad with two pieces of sarcoplasmic reticulum. You'll recall the sarcoplasmic reticulum is a big reservoir of calcium waiting to be released. It really wants to move out of the sarcoplasmic reticulum. Once the calcium is released from the sarcoplasmic reticulum, it bathes each of these myofibrils. These myofibrils are then going to be able to shorten because of the presence of calcium. So in the third stage, where we look at the sarcomere and crossbridge cycling, how the sarcomere actually contracts, we'll see the importance of that molecule of calcium. So, moving into stage one, let's take a look at this neuromuscular junction. We have the synapse of the neuron with the muscle cell membrane. The synaptic knob is where we'll start to see more calcium channels in the membrane of the synaptic bouton. When the signal arrives down the nerve at the synaptic knob, the calcium channels in the bouton are going to open, allowing calcium to move into the neuron at the bouton. This calcium will then attach to these vesicles of acetylcholine that exist inside the synaptic bouton of the neuron. The acetylcholine in these vesicles will then bond to the cell membrane and release the calcium out into the synaptic cleft. Once the calcium's in the synaptic cleft, it's going to cross the synaptic cleft and bind with acetylcholine receptors on the other side. Those acetylcholine receptors will cause the acetylcholine activated channels to open so that we can propagate an action potential along the muscle cell membrane, much like the potential was propagated down the nerve cell itself. So now that we have a signal delivered to the sarcolemma, or the muscle cell membrane, we need to see what happens as that action potential propagates along the cell membrane and down through the T-tubules. So now, we'll take a deeper look at that. You can see that we have our voltage-gated sodium and potassium channels. So an action potential is being propagated along the cell membrane. As it moves along the cell membrane and runs into a T-tubule, the signal is then propagated down the T-tubule. As the molecules move through the membrane and an action potential moves down the T-tubule, it excites the calcium channels on the sarcoplasmic reticulum. The calcium channels on the sarcoplasmic reticulum open, dumping tons of calcium out into the cytosol of the muscle cell. This calcium is then going to bathe all of those myofibrils, all of those protein assemblies that make the sarcomeres that eventually shorten. In our third step, we're going to look at how these calcium molecules actually activate the sarcomere to contract. So you can see crossbridge cycling is set up in, like it says, a cycle, where we have interaction of myosin heads with actin. Taking a closer look at really what's going on and using a little bit of an analogy, I'd like to show you how the sarcomere shortens before we look at the details of it. So we have, you'll recall, myosin filaments. We also have actin filaments. And we have our Z discs, we have the middle line, the M line, and we have various bands. And we'll take a look at those in more detail in some micrographs later on. But in order for this to shorten, the myosin filaments have to move along the actin filaments and draw these Z discs towards each other. The analogy I think of is it's much like rowing crew, where you have two rowing boats. The two rowing boats are going in opposite directions, but they're attached at the nose and the midline. So as the members, the rowers, have their oars in the water, they reach and the oars are attached to the actin and they row and they pull along those actin filaments, thus drawing the Z discs closer together. So imagine that these myosin heads are attaching to the actin and using a power stroke to draw the ends closer together. Now, it's time to really take a look at how calcium is involved in this whole situation, right? So, here we go. In the beginning, we have a molecule of actin. Now, there are a few more players on the molecule of actin. First of all, we're going to have tropomyosin. Tropomyosin is this long, green, fibrous molecule, and it covers up these little black dots, and those little black dots are myosin binding sites. It's where the myosin heads, the oars in our rowboats, really want to bind on to the actin molecule. However, before calcium is present, the tropomyosin covers up each of these binding sites, so they're not available for the myosin heads. So no contraction can happen if there's no calcium present. When calcium is present, though, it's going to bind to this molecule called troponin. Now, troponin acts like a little bit of a clip. It clips the tropomyosin onto the actin strand over the myosin binding sites. So those myosin binding sites are not available because the troponin clips are holding tropomyosin on. When calcium shows up, though, we have a different story. Calcium shows up, binds to the troponin molecule, and then causes the tropomyosin to move aside, exposing the binding sites so that now the myosin heads can attach. So let's take a look at that. The myosin heads attach, and then we'll note that there's also ADP and inorganic phosphate, which means that we've recently used some ATP, right? So the myosin head binds to the active site once it becomes available. And what we're going to see is that the ADP and the inorganic phosphate will fall off. And then, once those fall off, the myosin head can cock back. So remember with the rowing analogy, we had our rowers ready to go. And now we're seeing this power stroke. So we're moving a little bit closer to the Z disc on this end. The head now has been released, but it needs to get recocked. So somehow we have to have ATP involved. In truth, that head is actually stuck to the myosin binding site until ATP shows up. And it's only when ATP shows up that the myosin head can release and reset to make another power stroke. So once the ATP shows up, the myosin head is released. And at the same time, it's recocked into the ready position, right? The head is back. We have ADP and inorganic phosphate because we just used the energy of ATP to reload the head so that it's ready to make another power stroke. So again, this goes around in a cycle as the rowers in our myosin boats pull along the actin filaments towards their Z discs on either side. Remember, the noses of the boats are tied together, and the rowers are rowing in opposite directions towards the end of these, let's call them actin channels. The cycle continues until the sarcomere is completely shortened. So how does this actually look in the muscle? Remember that each of the muscle cells is full of myofilaments, and each of those myofilaments has many sarcomeres along it. Each of the sarcomeres is composed of actin and myosin. When we look at a micrograph, you can see here how the M-line Z discs are represented. You see these dark bands towards the center here. We call those the A band, right? And those dark bands result because of the overlap of actin and myosin. As the Z discs are drawn closer together, we see that there should be more darkness. So the A band is essentially the entire thing. There is no longer an I band where there is only one strand or the actin filaments involved. So essentially, as the sarcomeres shorten along the whole length of a myofibril, we see that the whole myofibril shortens. And if all the myofibrils in one cell shorten, then we see that whole cell shorten and thus contraction of a muscle. So now let's take a review of this whole process and see how it all comes into play to affect shortening of the muscle. Again, there were 3 stages. We had the neuromuscular junction. The next stage was excitation-contraction coupling, where we see that signal from the neural signal transfer across the membrane and propagate down the T-tubules. Recall that the T-tubules surround each and every myofibril, and that it's closely associated with sarcoplasmic reticulum. So sarcoplasmic reticulum is stimulated, the calcium gates open, calcium rushes out through the gates and bathes each of the myofibrils in calcium. And you'll recall that that calcium then binds to troponin, unclipping the tropomyosin, so the tropomyosin moves aside, exposing the actin binding sites. Those actin binding sites are then available for the myosin heads to bind to. Once the myosin head binds, the ADP and inorganic phosphate that were used from the ATP that cocked the head originally fall off, which allows the power stroke to happen. We're rowing towards those Z discs. Then ATP shows up. Not until ATP shows up can the myosin head be released from the actin filaments. But when ATP does show up, it releases the myosin head and uses the energy of ATP to cock it back and get it ready for another power stroke. Now we have, again, the inorganic phosphate and the ADP stuck to the myosin head until over here. So this cycle continues until the rowers in their myosin boats reach the end of the channel between the actin and touch the Z discs. Now we have a shorter sarcomere, a shorter myofibril, and thus a shorter muscle cell. So let me leave you with a couple of different questions. The first question I'd like to ask you is to think about the case of there being no ATP present. What if we've run out of energy and there's just no ATP? What happens to muscle fiber contraction? And then the second scenario I'd like you to consider is if we have calcium, perhaps there's not enough calcium. If there's not enough calcium to be released, what would be the effect on muscle fiber contraction? So when I come back in the next segment, we'll address those 2 questions and see what you've come up with.